The next speaker is Dr. Tricia Cottrell. She's also a thoracic pathologist. What? Try a little. She's also a thoracic pathologist at Queen's University and also a senior investigator at the CCTG. She's with the group at Johns Hopkins devised the immune response score, and now she'll be talking about immunopathology.
Thank you. Thank you for the invitation. I have no disclosures, and I have no disclosure slide. So I'm going to be talking today about multiplex immunofluorescence, which I think we've had a nice introduction to the concept of spatial profiling in the tumor microenvironment. I'm focusing today on pretreatment biomarkers of response to anti-PD-1 therapy. This is some older data comparing different biomarker modalities in terms of testing on a pretreatment biopsy and predicting response to immunotherapy. So this includes over 10 tumor types, over 8,000 patients, comparing PD-L1 immunohistochemistry in blue to tumor mutational burden in red, gene expression profiling in yellow, and multiplex immunofluorescence or immunohistochemistry in green. And we're looking at the ROC curve, which reflects the sensitivity and specificity of the association between the biomarker and the outcome, which in this case is response. And you can see in the top corner, there's a star at 1. That would be the performance of an ideal test. We never get that good. The PD-L1 immunohistochemistry, that is the current gold standard, is 0.66. And you can see that the multiplex immunofluorescence is performing much better than the other approaches with an AUC of 0.8. So the data that I'm going to share today is unpublished data from multiplex immunofluorescence analysis focusing on the neoadjuvant anti-PD-1 cohort that was published in Patrick Ford's original phase 2 trial. So we have pre-treatment biopsies as well as the on-treatment specimens. And then I'm going to show you a little bit of data from a couple other cohorts that were advanced metastatic, pre-treated non-small cell lung cancer that were also treated with anti-PD-1 as part of clinical trials. And I'm focusing today on data generated at Johns Hopkins using the AstroPath platform. So this was developed in collaboration with Janice Taub and Alex Saleh, and it really focuses on developing a very high throughput, high volume amount of data for the tumor microenvironment. So the first figure that you're looking at is actually a whole slide that's been covered over with individual high-powered field tiles. That's what all the blue squares are. And we image the entire slide and then actually stitch all of those images together and extract the single cell data from the entire slide. So you can see for the pre-treatment biopsies, we're getting thousands of high-powered fields for each of these cohorts, and then for the on-treatment specimens, which are much larger, we're getting over 30,000 high-powered fields, and so the total for this study is over 30 million cells. So this is a much greater amount of data than you get with a lot of the other profiling technologies, and one of the trade-offs for that is that we have a smaller set of markers. So in this slide, you can see a heat map showing the different lineage markers that are included in our panel, including CD163 for macrophages, FOXP3 for regulatory T cells, CD8. We're going to focus on this unique FOXP3 positive, CD8 positive population, and then we also have cytokeratin as our tumor marker. And across the top, you can see the expression of PD-1 and PD-L1 as assessed on each of these cell lineages. And when we look across the densities of all of these different cells and their subpopulations, and we look for correlates in the pretreatment biopsy that are associated with major pathologic response after neoadjuvant therapy, what we find is that this very rare population of CD8 positive, FOXP3 positive cells is the strongest predictor. And similar to that meta-analysis, you can see that the AUC values are in the 0.7 to 0.8 range with subsetting by PD-1 and PD-L1 status actually somewhat improving the prediction. Now, this cell population was previously identified as predictive of response to anti-PD-1 in melanoma in a previously published paper, and so we weren't completely shocked. We also have single-cell RNA sequencing data that I'm not going to show today that indicates that these are early activated T cells that have expression of CXCL13, which is suggestive that they are actually antigen-specific cells in the tumor microenvironment. The other thing I'll point out about these cells is that they are extremely rare. So you can see in that little table that they represent about 0.1% of all of those over 30 million cells that were profiled, and less than 3.5% of the CD8 positive T cells. So this is a population that would be very challenging to detect if you're using a subsampling approach. You really need to sample a lot of the tissue in order to find a rare signal like this. And when we look in our pretreatment biopsies for the neoadjuvant cohort, what we can see is that the densities of these CD8 FOXP3 cells actually correlate with patient outcomes. So the blue lines are patients who have high densities of these cells, and they have improved recurrence-free and overall survival, as compared to patients in the red line who have lower densities and worse survival. And then in one of the advanced non-small cell lung cancer cohorts where we had survival data available, you can also see that pretreatment densities strongly associate with the outcomes, particularly overall survival in this group. So at this point, we've seen that CD8 FOXP3 cells are predictive of IO response in melanoma. We're now seeing it in lung cancer. We're also seeing it across stages in lung cancer. So it has the real potential to be considered for a pan-tumor biomarker of response. We next wanted to dig into some of the spatial biology of this unique cell type. So we mapped the probability of these cells being located relative to the tumor stromal boundary. So that center line is the tumor stromal boundary. And what you can see is in gray is that the CD8 FOXP3 cells are actually most likely to be found just outside the tumor, and they're right along with the other CD8 T cells as well as the conventional regulatory T cells. In contrast, you can see the macrophages in the purple line are going right into the tumor. And this, again, was also demonstrated in the prior study in melanoma, and it also correlates with the idea that these are early activated T cells. They're encountering tumor antigen right at that tumor stromal boundary, so it makes sense with the biology that we think is happening. Next, we wanted to profile the contact neighbors. So you learned about all the different types of spatial profiling, and now I'm going to show some examples. And so what we did is we identified the cell lineages that were located within a 5 to 10 micron radius right around our CD8 FOXP3 cells. And what we did was we compared those nearest neighbors, the densities or the proportion of different cell lineages around the CD8 FOXP3 cell to the proportion of the different cell lineages across the entire tumor microenvironment. And we did this for all three of our cohorts. So again, cohort 1 is our neoadjuvant cohort, and 2 and 3 are advanced cohorts. These are all pretreatment biopsies. And what you can see in the darker color is that the CD8, in the dark yellow, the CD8 proportion is much lower in the background TME relative to right around the CD8 FOXP3 cells. And in contrast, you can see that the proportion of tumor cells is much higher in the background TME than it is immediately around the CD8 FOXP3 cells. And this pattern is consistent across all these cohorts. And so because it seems that these cells are existing in a consistent, predictable microenvironment or niche, we next sought to develop a model that could actually identify that niche even in the absence of a CD8 FOXP3 cell. And part of the motivation for this was that we know that the CD8 FOXP3 cells are relatively rare, and our hope was that if they were a surrogate for some kind of immune activity that might predict response, we might be able to get a stronger signal if we focus on their niche instead. And so what you see here in orange is the AUC curve for the CD8 FOXP3 cells, and what you see in blue is actually the AUC curve for the niche itself. And again, this is from the pretreatment biopsies in the neoadjuvant cohort, looking at prediction of major pathologic response. And in terms of abundance, what we found is that the niche, if you didn't require a central cell, was actually 150-fold more abundant in the tumor microenvironment than the CD8 FOXP3 cells. And so when you look at these ROC curves, you can actually see that the statistical error is plotted at both 68% and 95% confidence intervals, and the error is much higher for the CD8 FOXP3 cells, and we actually cut it down fourfold by using the niche as a spatial biomarker. So this is the type of process that we have to do to optimize these signals in order to make them as robust as possible for clinical translation. So in thinking about clinical translation, one of the big advantages of the multiplex immunofluorescence is that it's based on a technology that is very similar to what's already being used in the clinic. So we can stain slides on automated stainers that are almost identical to the automated stainers used in the pathology lab to do immunohistochemistry. And so this is a study that Janice Taub led across 6 different sites where they did a multi-site reproducibility study. And they stained at each of the sites serial sections of tissue microarrays and then compared the data. And what you can see is there's very high correlation for the signal that you get with the slides stained at the different sites. And you will notice that FOXP3 on the top right there is the one that has the most error. That's a more rare cell type, and because these are not the exact same slide, you do see a little bit more error when you're comparing serial sections. But otherwise, very strong correlations between these markers. They also demonstrated that you could reproducibly detect lineage-specific expression of PD-L1. So right now, when pathologists are interpreting PD-L1 in lung cancer, we're only looking at it on the tumor cells. And in other tumor types, you may look at it on tumor or immune cells, but not a specific subset of immune cells. So here we're looking at the reproducibility of detecting PD-L1 expression on a cytokeratin-positive tumor cell or a CD68-positive macrophage. And again, you can see nice inter-site reproducibility. Finally, they wanted to look at the reproducibility of the types of spatial analyses that we're interested in. So this is a proximity analysis looking at the density of PD-1 cells within a 25 micron radius of a PD-L1 positive cell. And again, you can see very nice correlations with these spatial analyses. So all of this is sort of building up to getting us to have these multiplex immunofluorescence and spatial biomarkers moving into the clinics. And so, the next steps in order to make this a reality are that we need much larger cohorts. I'll echo that request. And we really need this to optimize specific thresholds for these biomarkers. So that kind of data is going to require much larger studies. And then we need to get these into clinical trials, and we need to have prospective validation that these biomarkers are enriching for patients who are going to respond to therapy in order to advocate for them to be funded by payers and getting them into the labs. So that's all. Thank you.
I got one. Have you tried seeing if you could predict a cell is a FOXP3 CD8 positive cell off of an H&E?
No.
Maybe Logan can help.
Yeah. Logan, you want to do that?
Good idea. Tom?
Great talk. The CD8 FOXP3 positive cells, you're talking about the niche. Is the niche sort of a subset of a tertiary lymphoid structure typically, or what sort of immune niche are you looking at outside of just that CD8 cell?
Yeah, so they're not tertiary lymphoid structures. So the niche, it's kind of like the immune front that you're sort of seeing at that tumor stromal boundary. So, a lot of CD8 cells and fewer tumor cells. That being said, what we identified in the on-treatment specimens is that those CD8 FOXP3 cells actually do get into the tumor, and the densities of those CD8 FOXP3 cells do correlate with the number of tertiary lymphoid structures in the on-treatment specimen. So I think they're probably just 2 different markers of a functioning anti-tumor immune response, but they would be distinct. Tertiary lymphoid structures are hard in pre-treatment biopsies because you get so little tissue. there's just a high sampling error.
I would love to get into many more questions, but I think we're just running a bit behind, and